are Fireblighters and we are participating in iGEM Design League 2021 and this is our project.
Mexican crataegus, colloquially known as tejocote, is a fruit firmly involved in Mexican culture and traditions. For Puebla, it's an important import and export product. However, Erwinia millobora is considered the most serious disease in the tejocote trees and other Rosaceae family, causing havoc in the production of the fruit. As students from the state of Puebla, we realized the great problem that exists in the tejocote crops. So, on January 30th of 2020, we met with the tejocote farmers from this state in the municipality of Tlahuapan. These farmers belong to the organization from CESABEP, Puebla State Plant Health Committee, with whom an agreement was signed between our university and them to work together in favor of tejocote orchards. In addition to this, we gave them a workshop regarding how they have to clean their pruning tools. First of all, we wanted to learn from the problem of who is directly affected, so we could help them the best way, matching the needs of the farmers and the infected crops. The farmers explain how they approach to calm the expression of the pathogen, what are the products they use, how easily identify the expression of the pest in leaves, trunks, and fruits. And finally, they also explain how this pathogen affects their lives, and this motivated us to design an effective biocontrol system for this problem. We created a biocontrol strategy attacking the interior and exterior of the plant. The stigma surface of the flower acts as the major site of colonization for the pathogen, providing Erwinia millobora with nutrients in the stigma exudates and helping its growth. Raindrops are due allow it to reach nectarines through chemotaxis, which is used to actively enter the plant. So when Erwinia millobora is in the interior, it goes to the xylem, where it reaches a sufficient cell density in response to quorum sensing. The intervention in the exterior consists of reducing the colonization capacity in the pistil, causing Erwinia millobora to actively enter the plant with the motility gene. We are using the phage ENT90, which carries the ACPP gene in antisense orientation under control of the LuxPR promoter. It will sense the quorum sensing response with a co-expressed LuxR-AHL receptor and will block the essential production of the acyl carrier protein upon quorum sensing induction, and this will kill the bacteria. On the other hand, the interior strategy consists of avoiding the activation of its virulence genes, if it managed to colonize and enter anyway. We designed 4 different constructs that will be inserted into the filamentous bacteriophage PEAR6. 3 of them have the YHJK, JYDB, and EPSE genes. The difference resides in that the JYDB enzyme is targeted to different cellular compartments: cytoplasm, cell surface, and in the medium.
Inside local constructions were made according to the standard BioBrick RFC10, which uses a shortened prefix for protein-coding region so that the ribosome binding site is as close as possible to the ATG codon of the coding sequence. We also use the green fluorescent protein as a selection marker. The first construction includes the gene anti-ACPP, which will exert a bicidal effect on Erwinia millobora, but it is crucial that it is only expressed when necessary, so that it does not interfere with the bioreplication and reproduction. Therefore, in this plasmid, we are expressing as well the gene LuxR, a protein that forms a complex with the signaling compound homoserine lactone. This complex will bind to a palindromic site on the Lux promoter, increasing the rate of transcription of anti-ACPP so that it will only be overexpressed under quorum sensing response. In addition, we added stabilizing sequences in order to prevent the antisense RNA from degrading too quickly. Then, we introduced flanking sequences to this construction so that it will be introduced into the bacteriophage ENT90 by homologous recombination. The next 4 constructions were introduced into the PEAR6 bacteriophage, in which we used directed mutagenesis to introduce the restriction site of NOT1. The second construction includes the gene AMI-R, which under a non-native promoter produces changes in transcriptional levels of other genes, inhibiting the growth of Erwinia millibora. In order not to inhibit growth, which would produce an evolutionary advantage for strains that delete this gene, it is necessary that the protein is expressed only when it is required. Thus, we use the promoter looks in this construction as well. The third construction includes the gene EPSE, JYDB expressed in the cytoplasm, and YHJK. The fourth construction also included the same genes, but here the lactonase will be directed to the type 1 secretion pathway to the extracellular compartment. For this purpose, we created a fusion between HLYA and JYDB. In the fifth construction, we included the same genes, but this time we added the outer membrane protein of Enterobacteria, OMPA. The fusion OMPA-JYDB will allow the anchorage of the protein on the cell surface. If we succeed in reducing substantially the pathogenicity of Erwinia millibora, it will be a big win, maybe in the fight against this pathogen in other fruit trees too. But there will be still a long list of pathogens of the tejocote to be tackled before we can chant victory.
To prove the theoretical functionality of these enzymes to avoid the activation of the hormone sensing, a mathematical modeling was carried out that contemplates this entire process and how it would act in the presence of the enzyme. For this, the intracellular situation was analyzed separately from the extracellular one. For the intracellular, it contemplates the model for a single cell, while the extracellular one, it contemplates the number of bacteria present. These are connected with each other with the diffusion of the signal AHL and the enzyme. The model is then described by the following dynamic system. As mentioned above, the main improvements of this model with respect to the wild type is the incorporation of the enzyme. Having analyzed the work of iGEM Team Chihuahua, improvements were made such as a change to an enzyme with greater enzymatic efficiency and the use of real and not arbitrary enzymatic efficiency inside the model parameters. Also, the targeting of the enzyme to the extracellular space and the correction to mu to a real value and not arbitrary. It was changed from the enzyme AIIA to the enzyme JYDB, both acting for the same substrate. The enzyme proposed by Team Chihuahua, AIIA, from Bacillus sp, displays the following kinetic constants. While the one proposed by Team Puebla, JYDB, from Rhodocus sp, displays these. As can be seen, it works at 12 times lower substrate concentrations, is 15 times faster and therefore has a 200 times higher enzyme efficiency. This comparison can be seen in the following kinetic graph.
To reinforce our analysis, a biostability test was carried out to identify the amount of enzyme necessary to block the QS response, both in the intracellular and extracellular context. The analysis is based on the identification of stationary points within the system of equations, each representing either a maximum or a minimum. The new system of equation was obtained for a change of notation. With the new system, a graphic analysis was carried out to identify the solutions of the equations. From this graph, we observe that the system has 3 stationary points, and that it can be expressed inversely to observe the expression of the QS due to different concentrations. Therefore, this analysis was made for the 3 situations. From a number of bacteria of 2080, the QS response is triggered and can return to steady state with a number of 716 cells. At 4,000 cells, the peak concentration will be 0.158 nanomoles and it will never pass the 0.25 nanomoles. Using the AI enzyme proposed by Team Chihuahua, we observed that 3,015 nanomoles of enzyme will be needed to stop the QS. Therefore, the value of 650 nanomoles that they used wasn't enough. Using the JYDB enzyme proposed by us intracellular, we observed that only 16.5 nanomoles of enzyme will be needed to stop the QS. Using the same enzyme but extracellularly, we observed that only 9.7 nanomoles of enzyme will be needed to stop the QS. Next, we compare the graphs with the parameters for the 3 models, wild type, Team Chihuahua, and Team Puebla. There is an obvious reduction in the concentration of all the variables compared with and without our enzyme. The same case when comparing Chihuahua and Puebla models. Measuring the effect through time, we observe that the QS in a wild type will activate near the 15 minutes. For Chihuahua's model, the QS is only delayed and will need more enzyme. With our improvements, the QS completely stops, and in both directions of the enzyme, it will ensure the stopping of the QS regardless of the cell density. Using a more efficient enzyme also reduces the need for a strong expression and therefore avoids an evolutionary disadvantage from energy drain.
First, together with the student groups of Biotech Puebla and Women Plus Tech, which belong to the same university as the iGEM team, a panel of 4 women from different areas of engineering was held. Mechanical and processes, bioengineering, computational technologies, and applied science.
In this, a round table was held in which the participants answered questions about their personal experiences and also answered the doubts of the public. It was primarily focused on high school girls that wanted to or had doubts about pursuing a career in STEM. Although the general public was accepted, it should be noted that it had a reach of 21 people.
Subsequently, with the Linsen Bio, Biocontrol and iGEM 1 teams, a group of 4 experts in biosafety and bioethics were gathered to evaluate the proposals of each team in this area. So there was a feedback and improvement cycle, ensuring the delivery of a professional review document. For this, there were sessions between the experts and also between the teams.
The Scientific Sleepover was held in collaboration with the iGEM UAM and Biotech CCM teams. This was intended to inspire the students of 3 high schools to study a university career with an occasional focus on STEM. This was made through talks, lectures, experiments, roundtables, and open spaces for questions about the university. About the schools, the group interacted with the Lic Adolfo López Mateos High School of the State of Mexico for 4 days.
And with Prepa Tech of Mexico City and Prepa Tech of the State of Puebla, these last 2 are part of the same university as the iGEM team. In total, there were 82 registrations and 45 constant attendances. For these, each team contributed with part of the materials used during the sessions, and also there was daily interaction between the teams for a good coordination.
One of the activities that has the most impact on the team was that carried out with the student group and social organization Mateo Quinto, where the team carried out a well-fundamented and extensive research and also prepared a presentation during a whole week to conduct an hydroponics workshop, specifically about the strawberry, which was named Hydroponic Strawberry Cultivation Improvement Workshop. Mateo Quinto CSO is a non-governmental association that gives housing to children and adults while providing them with academic education, emotional, physical, and psychological support through economical aids and self-support by means of self-made products with their own resources.
The team executed a face-to-face workshop with the girls and women of the association about relevant topics regarding the care and cultivation improvements of the hydroponic strawberry.
Some of these topics were environmental factors that affect the plant, necessary nutrients for the cultivation, and typical plagues with focusing on Erwina vilobora. Performing as well activities during the presentation and receiving positive feedback from the women and girls to whom the workshop was given.
Afterwards, the team were headed to meet the greenhouses where such hydroponic cultivations were situated in order to identify pathogens and other non-favorable conditions that could affect the crops, so solutions could be proposed.
This part of the project is divided in 4 big sections. The first section is the project. In this part, we talk about the description, the objective, the analysis of the problem, and the viability. In this case, I will just add to the explanation of the video that the danger of Erwina vilobora resides fundamentally in 2 aspects: the practical impossibility of effectively limiting the diffusion and the lack of efficient control methods. So, for continue, we have the next 3 sections. The second, the regulatory framework, is where we describe the national for Mexico and the international framework applicable for this case. In the case of Mexico, it is important to stand up the General Law of Ecological Balance and Promotion of Environment, LEGEPA, and the project of Mexican Official Standard NOM 00 Zagarpa Semarnat 2015. The project also mentions other two general laws, but the LEGEPA and NOM are sufficient initial material in this area for the development of biotechnological products. At the international level, we have to highlight the Convention on Biological Diversity, CBD, and the Cartagena Protocol on Biosafety as the principal parameter generators for the biotechnological development. The third section is the security integrated to the project. In this area, we considered the next things. For consideration of in vivo experimentation, the DNA sequences will be obtained from the database and the iGEM parts. In addition, it is necessary that sequences and other oligonucleotides and plasmids need to be synthesized by. Therefore, the nature of the recombinant DNA sequences by suppliers. Due to regulation that exists with genetically modified organisms into the environment, the test will be done inside a greenhouse with controlled conditions instead of the open field. The testing in the laboratory will be in tejocote flowers or twigs with flowering and pieces of fruit. Therefore, the nature of the recombinant DNA sequences, vectors, and recipient organisms must be carefully evaluated, as well as any potential biohazard associated with experimental settings. Phages as well as endemic bacteria, Erwinia millivora, handled in laboratories that are engineered to present no biological risks to human health can be handled in BSL1. To finish, as fourth section, we have a proposal. Here, the safe and security area describes the regulation by the necessity of the creation of a new NOM, or in the other hand, the modification of the existing NOM mentioned in the project. This as a new specific guideline that allow us to regulate the operation of the biocontrol system, taking into account everything related to the development, security control, and final disposal of elements. Also, we have in this section the vision for the application, where the Semarnat is the principal actor, of course in the company of other federal and local state actors are shown in the graph, so that can apply all the necessary public policy considering the Sustainable Development Goals like 2, 8, 9, and 13. These for conquer the next 4 big results. 1. The dissemination of the information generated. 2. The subside application of this biocontrol system. 3. The development of a normative and commercial ecosystem. And 4. The development of committees that allow constant evaluation of the biocontrol system, considering principles such as bioethics, the sustainability, and the social justice.